Brain lesions. Localizing areas of damaged or destroyed brain tissue have been instrumental in elucidating the functions of specific brain regions. By selectively targeting particular areas, researchers can observe the resulting behavioral deficits and draw inferences about the role of these regions in various cognitive and motor processes. This technique, known as lasering, has been employed for centuries, dating back to ancient Egyptian and Mesopotamian practices. Methods of producing brain lesions. These are several methods used to produce brain lesions in animals and, in some cases, humans. These methods can be broadly categorized into surgical, chemical, and electrical technique. Surgical ablation. Aspiration. This involves using a fine tipped stick to physically remove a brain tissue. It is often used to create relatively large lesions. Excision, a more precise method that involves cutting out a specific area of brain tissue using a scalpel or similar instrument. Chemical ablation, neurotoxins. These are substances that are selectively destroy neurons. Example include ibonitinic acid and cyanic acid and 6-hydroxydopamine. Radio frequency license. This involves using heated rock to destroy tissue. The heat can be generated by electrical current or by microwave radiation. Electrical ablation. Electrolytic lesion. This method involves passing an electric current through probe to destroy tissue. This intensity and duration of current can be controlled to create lesions of varying sizes. Considerations in producing brain lesions. When producing brain lesions, several factors must be considered. Specificity. The goal is to target a specific brain region while minimizing damage to surrounding areas. This requires precise surgical techniques or the use of selective neurotoxins. Second one is the size. The size of the lesion can influence severity of behavioral deficits. Smaller lesions may result in subtle deficits, while larger lesions can lead to more severe impairments. Location. The location of the lesion is crucial. Targeting different brain regions can produce vastly different behavioral outcomes. Animal model. The choice of animal model depends on the research question. Some animals, such as rats, mice, are more commonly used due to their relatively small size and ease of handling. Ethical consideration. The use of animals in research, ethical concerns. The researchers must adhere to strict guidelines to ensure the human treatment of animals and minimizing suffering. Application of brain lesion studies. Brain lesion studies have provided valuable insights into wide range of brain functions, including motor control. Lesions in motor cortex region can lead to paralysis or deficits in movement sensation. Damage to sensory cortex region can impair sensory perception, such as vision or hearing. Cognition. Lesions in frontal lobe region have been linked to deficits in executive function such as planning, decision making, and problem solving. Emotion. Damage to structure like amygdala can affect emotionally processing and behavior. Memory. Lesions in the hippocampus and other brain regions have been implicated in memory deficits, including amnesia. Producing brain lesions. Immediately underneath the skull, anesthetic animal cut scalp, remove the part of skull, cut through the dura mater, use suction device peptide to remove the brain material for deeper brain tissue. This method use electrode to burn away the tissue, very gentle, destroys all surrounded tissues and cells. Excitotoxic lesions, a tannic acid kills neurons by stimulating them to death. However, passing an electrode through or injecting a chemical into brain tissue can destroy other tissues along the way. So, next is the stereotaxic surgery. It is a brain surgery using stereotaxic apparatus to position an electrode or cannula in a specified position of the brain. In order to predict the location of the brain area, use stereotaxic atlas and use Pregman, the junction of sagittal and coronal structure of the skull, as a reference point. And stereotaxic surgery is one of the method to study the brain structure. Despite the limitations associated with the studies, including the potential for unintended damage and the difficulty of controlling lesion size and location, this technique remains a valuable tool for neuroscience research. By systematically studying the effects of brain lesions, the scientists can give a deeper understanding of the neural basis of behavior and cognition.